My name is Jan Grau. I'm from Halle University, Germany, and I will briefly walk you through our poster entitled Widespread Defects of DNA Methylation and Intra-motif Dependencies Revealed by Novel Transcription Factor Binding Models. And this project has been joint work together with Florian Schmidt from Genome Institute of Singapore and Marcel Schulz from Frankfurt University, Germany. The basic idea of this project has been to evaluate the effects of DNA methylation on transcription factor binding based on in vivo ChIP-seq data using dependency models. We already know that DNA methylation may affect transcription factor binding, and we wanted to investigate how well such effects could also be detected from in vivo data. It has also been shown previously that dependency models may more accurately reflect transcription factor binding landscapes. And we expected this effect to be even stronger when considering DNA methylation, since methylation itself is context sensitive. Our general workflow is as follows. We obtain whole genome bisulfite sequencing data for a cell type or tissue. Within the genomic sequence, methylated Cs are then converted to M, and Gs opposite of methylated Cs are converted to H. In addition, we collect ChIP-seq data for the same cell type for different transcription factors, or in the following short TFs. And then we perform de novo motif discovery from the sequences under the ChIP-seq peaks using the methylation-aware genome variant. The motif models we consider are first the traditional position weight matrix model. Second, we consider SLIM models, which are capable of capturing intra-motif dependencies. Regarding data, we collect whole genome bisulfite sequencing data from ENCODE and DEEP. For the ChIP-seq data, we obtain peak files for the same cell types from ENCODE. Here we do not filter for TFs containing a CpG in their binding motif in advance, which also means that a fraction of the TFs we consider may not be affected by DNA methylation. To test whether considering DNA methylation or using dependency models or a combination of both leads to more accurate binding models, we consider this a classification problem. And we measure performance by the area under the precision recall curve in a 10-fold cross-validation. So in total, we had 4 modeling alternatives, considering DNA methylation or not, and using PWM models or dependency models. Briefly, we find that for 54 of the 143 TFs considered, the combination of dependency models and methylation information indeed yields a significantly improved performance compared with the PWM model using the original genome. For 27 cases, we also find that dependency models add to using PWM models on the methylation-aware genome variant. And for the same number of cases, we find that methylation information further adds to only using dependency models. So let's look at a few examples of TFs for which considering methylation proved to be especially beneficial. In the upper part of each plot, you see a sequence logo of the predicted binding sites. In the lower part, we further represent the abundance of CpGs in these binding sites per position, and you find a measure of methylation sensitivity, which basically represents what the model learned how binding is affected by DNA methylation. If these values are negative, like in these 3 examples, this means that the model thinks that methylation is detrimental for TF binding. And this is a pattern we find in general among the TFs we studied. DNA methylation is mostly detrimental for binding. We also find that the effect of DNA methylation may differ among TFs from the same family. Here we show results for FOXA2 and FOXK2, and we see that the pattern of methylation sensitivity differs. FOXK2 has two positions, which may be CpGs in some cases, and these are more strongly affected by methylation. If we had not taken the unbiased approach initially, examples like this might have escaped our attention since neither motif contains a prominent CpG. Finally, let me show you one example where the importance of dependency models can be illustrated briefly. Here we show a dependency logo of the predicted JUN-D binding sites. Binding sites of JUN-D may be divided into two half motifs, TGA and TCA. And the spacer between those two halves may be 1 or maybe 2 nucleotides. What we see from this dependency logo is that of the variants with a short spacer, only a small fraction of binding sites contains a CpG, and this is only slightly affected by DNA methylation. By contrast, for those with a long spacer, we see a much stronger effect of DNA methylation. Both variants could not be captured well by a single PWM model, but using dependency models, this is easily possible. If you got interested in trying such analyses for your own data, our tool Medemo is available under this URL. And it comes as both a command line interface version and a graphical user interface version, which can be used for motif discovery, but also for generating plots like you find on the poster.